Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Afif bin Alias. Hello, Sir Amir. Today I will be presenting about life cycle overview. This is just an overview, which the life cycle in details will be presenting by Joshua. So, let's start. When plant reproduce sexually, they undergo alternation of generation, which they alternate between two multicellular stages, which first is haploid and diploid. So in flowering plants, diploid sporophyte is dominant and it is the portion of the life cycle that bears flower. So a flower is reproductive structure of angiosperm, which it will produce the two type of spores by meiosis, which were megaspores and microspores. So, okay, next slide. So the spores will divide mitotically to become haploid gametophyte. So haploid microspore undergoes mitosis and becomes a pollen grain. Pollen grain is male gametophyte, and this pollen grain is either wind-blown or carried by an animal to the surrounding of the female gametophyte. So haploid megaspore will undergoes mitosis and becomes female gametophyte, which was embryo sac. So this embryo sac is within the ovule that is within an ovary. At maturity, a pollen grain containing non-flagellated sperm will travel by way of pollen tube to the embryo. So apabila this sperm fertilize an egg, it will produce a diploid zygote. So the zygote becomes an embryo, but it is still within an ovule. So this ovule will develop into seed that contain the embryo and also a store food surrounded by seed coat. So this seed will germinate and the new sporophyte emerge and through mitosis and grow become the diploid sporophyte, which becomes a matured organism. So as you can see here, this is the life cycle of sexual reproduction in flowering plants. As I already explained, this is reproductive structure. A flower reproductive structure will produce two types of spore, microspore and megaspore. This microspore will become male gametophyte, megaspore will become female gametophyte, and in this egg we fertilize the sperm to to produce the zygote. The zygote will become an embryo and and this embryo is within an ovule. The ovule will develop into a seed that contain a bryo and store food surrounded by seed coat. So the seed will germinate a new sporophyte through mitosis. Through mitosis, the seed will germinate through mitosis and grow become a matured organism. That's all from me. Thank you.
Hi, my name is Mirza Shazina and my matrix number is ES10461. Today I'll be presenting about flowers. So flowers is a seed-bearing part of a plant. It consists of reproductive organs, which we will cover soon, and it is surrounded by brightly colored petals, which is known as corolla, and also green sepals, which is also known as calyx. Flowers develop when shoot apical meristem stops producing leaves and start producing flower enclosed with bud. Flowers are not only pretty, but they also have few functions. So firstly, the first function is flowers produce spores. So spores are tiny organisms that actually contain only a single cell. So next, flowers also protect the gametophytes. Gametophyte produce sperm cells and the female cells. So for like an example, the male gametophyte is the pollen grain and the female gametophyte is embryo sac. So next, flowers also attract pollinators. Flowers attract pollinators. Pollinators are animals that move the pollen from anther to stigma. Anther is the male part of the flower, and stigma, you can say it is the female part of the flower. So the most common example of pollinators are bees. A term where we call flowering. So flowering is a response to environmental signals such as length of the day or time of year. So next we will move on to the flower structure. So the first structure is sepals. As you can see, this is the sepal, and it is the leaf-like structure of the flower. It is mostly green, and the function of the sepal is to protect the bud as the flower develops within. Sepals are also known as the calyx. Next, we're gonna look at the petals. This is the petal, and colors of the petals is what makes the flower attractive. The size, shape, and color of petals are attractive to specific pollinators only. So petals are also known as corolla, as I mentioned earlier in the introduction of the flowers. So this is a fun fact. Wind-pollinated flowers have no petals. I'm gonna move on to stamens. Stamens is this part, and it is the male portion of the flowers. It consists of two components, which is the anther and the filament. So filament is the stalk, and anther is the sac. Like anther is supported by filament. And then next, we're gonna see the carpel. Carpel is the female portion of the flowers. It consists of stigma, style, and ovary. So the style is the stalk, and it supports the stigma. And then the ovary, they enclose one or more ovules, which is this one. So ovules, they produce megaspores. And so that's all for flower structure. Next is the variation in flower structure. We're gonna look at the variation in flower structure. So when a flower contains both stamen and carpels, they are called the perfect flowers or bisexual because they have both stamen and carpels. But when flowers only have stamen or only carpels, they are called the imperfect flowers or also known as unisexual. So other than that, we can also see that when both staminate and carpellate flowers occur on a single plant, they are called the monoecious. Mono means two, so both staminate and carpellate flowers occur on a single plant. Staminate is the male flower and carpellate is the female flower. But then when both staminate and carpellate occurs on separate plants, they are called the dioecious. Di means two, so both staminate and carpellate, the male and female, occur on separate plants. The top picture here is a flower called jatropha. This is an example of monoecious plant because they have both carpellate and staminate in the same plant here. So, and this one, the below one here is called the holly trees, and this is an example of dioecious because they have both staminate and carpellate on different plants. The berries are produced by the female plant, the fruits, and then the pollen are produced by the male plant. So that is all for my presentation. Thank you.
Good day to Usame and everyone watching this video. So today I'll be explaining about the life cycle of angiosperms, also called as flowering plants, in detail. But before I start with my presentation, I would like to give us a small information that our group will be preparing a set of questions which could be used also as your notes for your future. in your future for studying this particular subtopic. So let's start. Okay, so before I go into the topic, I would like to explain two important terms, which are sporophyte and gametophyte. So I think I think you all should remember these terms because we studied in the last semester. Sporophytes are multicellular diploid phase, is the multicellular diploid phase, whereas gametophyte is the haploid multicellular phase. Both of them are the most important or the most basic components in the life cycle of angiosperm because it moves on like this: sporophyte to gametophyte, gametophyte back to sporophyte, sporophyte back to gametophyte, and this whole thing goes on again and again for the next generations. So sporophytes, which are diploid, produce haploid spores. So haploid spores then eventually develop into haploid gametophyte, and these haploid gametophytes are the one producing haploid gametes, which would fuse to form a new organism a new diploid organism in the time to come. So basically, flowering plants are heterosporous. Heterosporous, the name itself says everything, which means it has more than one spore. So there are two types of spores present in flowering plants, which are megaspores and microspores. Megaspores and microspores. So basically, megaspores are usually will become mature female gametophytes, whereas microspores become mature male gametophytes such as the sperm cell. Okay, let us move on to the next part, which is a brief explanation on the development of male gametophyte and female gametophyte. Okay, this is the whole process in one diagram. You can find it in Mather book, page 296. Yeah. Okay, so development of male gametophyte. So male gametophyte develops in the male reproductive system. So what is the male reproductive system? Consists of two main organs, which are anther and the filament. So the development of pollen grains or the male gametophytes happens at the anther of the plants. So the anther produces microspores. As I said before, microspores are the ones needed for the production of male gametophytes. So these microspores are formed in the pollen sacs of the anther. So the anther has all the anthers usually have 4 pollen sacs, and these pollen sacs produce a lot of microspores. And these mother microspores, or the mother microspores, also known as diploid microspores, undergoes meiosis to produce 4 haploid microspores. So these haploid microspores, each of them has one nucleus respectively, but with half the number of chromosomes, which is suitable to be formed as a gamete. So every one of these nucleus would undergo mitotic division to produce 2 identical nucleus. But somehow something weird happens here. Unequal cytokinesis. I think you know what is cytokinesis, which is the splitting of the plasma membrane, splitting of the cytoplasm and everything. But unequal cytokinesis is where one cell formed would be bigger, the other cell formed would be smaller. This is very important and necessary for the development in male gametophyte because both cells formed, though they have the same nucleus, but they are different cells, where one of them is the tube cell, which forms the pollen tube after germination, and the other one is the generative cell, which forms the 2 sperm cells required for double fertilization. I'll explain about double fertilization later. It's a very important term. Okay, the 2 sperm cells important for double fertilization after germination of the pollen grain. So these 2 cells, tube cell and generative cell, which have different sizes, usually the tube cell is bigger than generative cell, they are enclosed in a membrane, and this whole cluster is called a pollen grain. So pollen grains are basically immature male gametophytes with these both cells. So why it is immature? It gets mature only when it gets germinated, or only when it goes and falls on the stigma of a female reproductive system. Okay, let's move on to the next part. Okay, this is just a simple microscopic picture. Okay, next. Okay, female gametophytes. So how are female gametophytes formed? So female gametophytes are formed in the female reproductive system, which consists of the stigma, the topmost layer of the female reproductive system, followed by the style and the ovary, which consists of 1 or more ovules. So 1 or more ovules. These ovules are the most important parts in this chapter. So ovules consist of 3 main parts, which are the parenchyma cells, which is inside, integuments, which usually covers the ovule, which is thick, which are thick cells, and also the other one is actually the micropyle, which is the small opening at the bottom of the cell. Okay, so what happens in the formation of female gametophyte, the development of female gametophyte? Okay, parenchyma cells undergo growth and they grow into megaspores, which is the mother cell, which is diploid. Megaspores are diploid. I repeat, mother cell megaspores are diploid. But after it undergoes meiosis, it produces 4 haploid megaspores. So these 4 haploid megaspores are half the chromosomal number, which also aids in the formation of gametes at the end. Among these 4, shockingly, only 1 gets useful. The other 3 is like, okay, you can just throw it away. Okay, so only 1 megaspore is becoming functional, and inside this 1 megaspore, there is only 1 nucleus. But somehow, this 1 nucleus undergoes mitosis in a certain manner and produces 8 new identical nucleus. Though 8 identical nucleuses are produced, but after the formation of cell walls, only 7 cells are found. Why? You may wonder. 8, 7, it does not tally, right? Okay, the whole story is one of the cells is binucleated. So what is binucleated? Binucleated means that there are 2 nucleuses in one particular cell. So what is a female gametophyte consist of? It consists of 7 cells with 8 nucleuses. You can count here 1, 2, 3, 4, 5, 6, 7, 8, and 7 cells 1, 2, 3, 4, 5, 6, 7. Okay, so what are the cells found inside here? Egg cells, the first and most important thing in among all the other cells, the egg cells, supported by 2 synergids, 3 antipodals, and 1 central cell, which is also important in the formation of endosperm. Okay, so what happens next is the development of new sporophyte. So how is new sporophyte developed? New sporophytes are developed by the process of fertilization, double fertilization. This is the most important term in the subtopic, double fertilization. I'll explain to you why it is called double. But how an enter from a specific male reproductive system gets transferred to a female reproductive system? It is transferred through the process called pollination. Pollination is the transfer of pollen from an enter to another stigma, from an enter to the stigma of a carpel. Carpel is the actual name for female reproductive system. So there are 2 forms of pollination, which are self-pollination and cross-pollination. Self-pollination occurs among the same plant, whereas cross-pollination occurs among different plants, but of the same species. So a mango, for example, a mango tree and a mango tree, not a mango tree and a coconut tree. It does not match, okay? Okay, moving back to our topic. So what happens now? Pollen grains from the anther goes and land on the stigma of a carpel or a female reproductive system. So what is happening now? I mean, pollination, either self or cross pollination, pollen grain lands there, and this pollen grain gets germinated. So as I said before, I hope you remember, when this gets germinated, the pollen tube forms, and this generative cell would produce 2 sperm cells, and this aids in double fertilization. Okay, so the formation of pollen tube causes the pollen tube to go through the stigma, following the style, and reaching the micropyle. So once it reaches the micropyle, what happens is that it bursts. And when it bursts, this generative cell, which has actually became 2 sperm cells, enter the ovule. So once it enters the ovule, since there are 2 sperms, it has to bind with 2 cells. So one of them, as you expect, as you can guess, it binds with the egg because it has to form a new baby. Joking. It has to form a new seed, okay? And the other one would bind with the central cell, which has 2 nucleus. So this would cause a 3N to be formed, and this 3N is called the endosperm nucleus, and this endosperm nucleus would form the endosperm. So endosperm is the nutritive tissue formed by endosperm nucleus as an energy source for the sporophyte. So this provides energy for the sporophyte to survive. And on the other hand, the sperm and egg bind to form a seed, and the seed development part will be explained by my partner in a short while. Meanwhile, the part of pollination, the cross-pollination subtopic, will be explained by my friend as well. So in short, I would like to conclude here, the most important thing in this subtopic, double fertilization process occurs between 2 cells, one egg and sperm. Another one is the central cell and the egg, forming the endosperm and also the seed. And another few things you need to consider is the production, is the male and development in male gametophyte and female gametophyte. And through this, I think you can grasp this chapter easily. And for your extra reference, I would be providing the PDF folders soon. So that's all from me, and I hope you all could understand. If you could not understand, you can refer to this diagram for an easy understanding. Thank you.
So we talk about the cross-pollination, which consists of two types, is wing pollination and animal pollination. Okay, some species of flowering plants such as grass and greens rely on wing pollination, as do the gymnosperm. Okay, much of the plant's energy goes into making pollen to ensure that to ensure enough amount of pollen grains reach the stigma. This one is animal pollinators. Insects, birds, mammals help in pollination. So most angiosperms rely on animals such as insect, for example, bumblebees, flies, butterflies, and moths. For birds, example, hummingbirds or mammals, uh, such as bats, to carry out pollination. So the use of animal pollinators is unique to to flowering plant, and it help those plant are successful on land. So as we can see, this is plant and their pollinator. So for millions of years, plants and their animal pollinators have co-evolved. So what is the meaning of co-evolve? Co-evolve is as one species change the other species undergoes adaptation in response, causing both to suit one another. So plants with flower that attract a pollinator had an advantage because in the end they produce more seed. Similarly to pollinators that were able to find and remove food from the flower were more successful. So this is example of plant and their pollinator. As we can see, this is bee and west pollinators flower, which the bee pollinated flower tend to be yellow, blue or white because these are the colors bee can see, which these respond to ultraviolet marking called nectar guides that help them locate nectar. So the color that bee cannot detect is red color. So other example is this butterfly pollinated flowers and birds and bat pollinated flowers.
Hello everyone, I'm Fatimi Shahira from Shahabudin. My metric number is AS10421. Today I will continue present our last subtopic, which is the seed development. Let me show you the slide. Okay, this is the seed development part. Okay. As you all know, okay, this is the seed development. What is the development? In this term, we call it the process series of stage from the simple to the more complex form. This is like our body. We know that we came from the cell, then tissue, then the organ, and then the system. We all know that. Like seed, it also seems like us. Usually it's from the simple to the complex one. It will including the differentiation and the specialization, which is their own, like us, our own organ have their own function. Same like this seed. All the components have their own specialized function to do. Okay, let's go to the seed development. Okay. There I have 4 seed development stage, which is the first one is zygote embryo. Next is the globular stage. And the third one is the heart and torpedo embryos. And the fourth one is mature embryo, which will have the seed already. Okay, this is the initial show about the seed development, which is first we have the fertilization, which after the fertilization, start with the first stage and go to the second stage with the globular, third stage, and lastly will be the complete seed. Okay, next we go to the first stage, which is zygote and proembryo stage. Yes. Then after double fertilization, zygote and endosperm become visible, which is we can see in this picture. This is endosperm and this is zygote. This is after fertilization. The zygote and endosperm become visible. So next, the zygote small with denser cytoplasm, which is the cytoplasm. This, you see this? The red one, okay, denser cytoplasm, and this is the zygote. Okay, this is the clear one presentation. Okay, this is the fertilized egg, and this is the apical cell and the basal cell, which is this zygote. Next, the zygote divided repeatedly, and it will become the proembryo. Okay, the zygote. Okay, this is the zygote, and it divided repeatedly. See this? And the elongated structure we call this like this. Okay, we see this is the zygote and have the apical and basal, and then in the middle of it have the elongated structure, which is we call it the suspensor. The function of this suspensor is to produce and transfer nutrients from the endoplasm to the seed. Okay, you see, this is the egg, and then we have the between to be the proembryo, which is after divided repeatedly, get the suspensor. Then we go to the next stage, which is globular stage. Yes. Okay, the globular stage is the proembryo, largely a ball cell. Why is it called ball cell? Which is this, the globular stage. You can see the zygote is to be a ball shape, in the ball shape. Okay, next, the root shoot axis of embryo already established, which is root shoot. Is this the upper one is the shoot, and the lower one is the root, and this axis have already established in this stage, which is the globular stage. And then the embryonic cell near the suspensor go on. Embryonic cell near the suspensor, which is the upper one, will go on, which is the root. This one, the basal cell will go on and to be a root. And the other and ultimate become shoot. Other one is the upper one, which is the ball cell, will go to be a shoot. And then the outer cell, dermal tissue plus cell plate will become a single outer layer, which is the outer layer, this one, the outer layer. Next, we have this, the globular, and then we will go to the triangular is the process between the globular and heart stage. Next, we go to the heart and torpedo embryos, which is the step 3, the stage 3. Okay, the embryo has heart shape, which we can see in this picture. Is the heart shape, and it is suspensor. Okay, next, which is the cotyledon or seed leaf. This is rapid cells division in this, and rapid cell division to become embryo continue to enlarge and elongate, and after the embryo continue to enlarge and elongate, it will become a torpedo shape, which is this shape. You can see it start to show two cotyledons, which is this is a eudicot stage. See. Okay, now the underground will go, which is we call the root apical meristem. We will go to become a root, and the shoot apical meristem here it will go to the shoot, and ground meristem will give rise to part of the embryonic. This is all the ground meristem. Next, we go to the last stage, which is the mature embryo, and this the epicotyle. Where is the epicotyle? Is the portion between the cotyledon to shoot development, which is this epicotyle. Yeah, between the portion between the cotyledon to the shoot development, which is should become from here, the epicotyle. We have this picture. This is epicotyle. For shoot development, this epicotyle. This and this epicotyle. Then the plume from tip of the epicotyle consists shoot tip and hair of small leaf. Wow. Like, you can see this is the one. It should be here for the shoot, have the tip of shoot. And then we have hypocotyle, which is the between the cotyledon to stem development, and termination, which is the epi is up, which shoot development, and hypocotyle is down for root development to stem development, which is this one. For the stem development, hypocotyle is for stem development. Or the embryonic root also from there. Okay, that's all. For the first stage we have known in the seed development, which is the first one is the zygote embryo stage, which is zygote is after the fertilization we have the zygote, and then the zygote will go the division and divide effectively and become a pro-embryo, which is the elongated structure we call the suspensor. Okay, next we go to the globular stage, which is the ball shape. Remember the ball cell. And then we go to the heart and torpedo embryo, which is we will become a heart shape, and then we'll go to the torpedo embryos, which is this torpedo will become two cotyledon. If it is a eudicote, see, and then lastly it will become a mature embryo. Okay, let's continue. So the difference between the monocot and eudicote cell seed, which is we all know that monocot, which is they have a one cotyledon. What is the cotyledon? This is the cotyledon. This is the cotyledon. This is the cotyledon. They have only one, which is this. The example is corn seed, which is this is the monocot seed. They have only one cotyledon, which is this cotyledon, and this is the endosperm. Next, the eudicote means have two cotyledon, which is 1 and 2. Next, monocot have only one seed left inside the seed coat. It is often only a thin leaf because the endosperm to feed the new plant is not inside the seed leaf, which is the monocot have only one seed left in their seed, which is the eudicote have two seed leaf. This inside the seed pod. They are usually round and fat because they contain the endosperm to feed the embryo plant. This endosperm is not inside the seed leaf; it's outside. Okay, next is in the cotyledon seed. Have the cotyledon contain certain nutrient and absorb other nutrient from the endosperm embryo, which is this. The cotyledon contains certain nutrient, so they need to absorb nutrient from outside for their endosperm nutrient. Then the eudicote have the cotyledon contain all nutrients, so they do not need to absorb from endosperm, which is why the endosperm big in the monocot seed. Okay, we have now three different between the monocot and eudicote seed. First, we have a one cotyledon for the monocot, and eudicote have two cotyledon. And then we have about the seed leaf, and then we have about the nutrient, which is the monocot have certain nutrient and need to absorb from the endosperm, and then the eudicote have the cotyledon have contain all the nutrient. And here have two links about the process of the seed development, so you guys can watch it for more explanation for the real one. Okay, that's all from us. Thank you. That's all from me. Thank you.